Welcome to my channel. Let's talk about the basic principles to understand diagnostic test results. I'm using the COVID-19 example. It's July 2020, and I get a lot of questions. Should I get the nasal swab, the blood test? So I thought it would be good to have a simple explanation on what these diagnostic tests are. I started by drafting 8 questions, so feel free to pause this video and you can take a look at the 8 questions. You can do a print screen and try to answer them. And then I will be answering them as we go along, and you will see how correct or incorrect you may have been and understand better the concept by understanding the answers to these questions. If we take a look at the virus COVID, for COVID-19, which is the SARS CoV-2, we see the structure that by now everybody has seen many times. It is an RNA virus, which means that it has the genomic material is based on RNA, not DNA. So this RNA is encoding for several proteins. And the proteins that encodes are the spike protein, which is what we see here in the schematic from the Encyclopedia Britannica that I'm using, is depicted in red. This particular spike protein is what gives the coronaviruses their name because it looks like a crown, but it also is the piece of protein that allows them to bind the host cell, and that's how they can infect the host cell through the spike protein. It also contains a membrane protein because the RNA has to be protected. There is a membrane, and of course there's the protein in the membrane, which also has lipids. It also has the nucleoprotein, which goes around the RNA, protecting the RNA from degrading or denaturing. And it also has an envelope protein. In addition to these 5 proteins, it also contains RNA-dependent RNA polymerase, which was described in Science in 2020. It is crucial for this virus to contain its own RNA polymerase. It will produce more RNA based on RNA. Normally, the RNA polymerases that we have produce RNA based on DNA. But in RNA viruses, they have to be RNA to RNA. And of course, these are the 6 proteins that are characteristic of coronavirus. In this particular example, published in the Proceedings of the National Academy of Sciences in June of 2020, there's a very elegant work done that describes the 6 genomes that are conserved throughout the coronaviruses. There's many different family members of this genus of viruses. And these 6 proteins are conserved in the genomes. What they did was to do an integrated analysis. It's a very good paper. I encourage you to read it. And they actually identified which were the key genomic features that could differentiate the more pathogenic clades from the less pathogenic coronaviruses. So even though these 6 are present in all 6, what are the pieces that differentiate them so we can understand differences between them? In this particular talk, I'm going to focus on diagnostic tests. So how can we actually find the ones that could help us make the accurate diagnosis? Looking at the structure of the coronaviruses and the different proteins that they have, it is important to remember why we need to understand the structure. So these particular 2 proteins, the spike glycoprotein and the RNA-dependent RNA polymerase, are critical for the virus to infect and produce disease. The spike protein is the one that binds to the host cell and allows the virus to infect the cell. So these are very good proteins for targeting them as antivirals. If there's an antiviral that can destroy or interact with that spike protein, the virus will not be able to infect the host cell. Also, the RNA-dependent RNA polymerase, if there's an antiviral that can tackle that particular protein and denature it or prevent it from being able to produce more RNA, even if the virus gets in the host cell, if the antiviral treatment can interfere, then the virus cannot replicate. So these two are very important proteins for potential targets for treatment. But when we're talking about diagnostic tests, which are the proteins that will help us, or the RNA that helps us identify which is the virus that's infecting the person? It is described that the spike protein is the one that would be more useful when we find it to identify not only the presence of coronaviruses, but also there are regions in the spike protein, special pieces of them that are very specific for one or another. And in this case, for the SARS-CoV-2, there's a specific piece or genome within the S protein that seems to be quite specific for it. So if I want to make sure that I can diagnose if the person has that particular coronavirus, it is best to focus on looking at that particular RNA. So let's talk a little bit about the DNA, the genes, and how they work. The double helix has been well known for many, many decades and well understood. We have one strand of the DNA is the one that is the encoding or the coding strand. This is the one that has the sequence of the nucleobases of DNA that provide the coding for translation to proteins. The other strand of the DNA does not have any coding. It basically is a complementary strand that has no code, and its whole function is to protect the genetic coding, basically just like a cover or a lid of a container. What the lid is covering is protecting it, but what really we care about, or what really is going to be translated, is what's in the coding strand. The structure of the DNA has phosphate sugar, and it's basically on the two ends on the outside. This is sort of the structure and is protecting the inside, which has the coding message. And that is basically created by having the four different nucleobases. It's cytosine, guanine, adenine, and thymine. And if you can see on the right, they have a way of always combining adenine with thymine and guanine with cytosine. It always combines that way through the molecular structure. When we move to the message RNA, first thing that I want you to notice is that the nucleobases and the sequence of these bases mimics the non-coding DNA, because the idea is that it will, by having the translation into proteins, it has the non-coding brand because it will be read as the coding brand. So the coding gene is not the one that is mimicked by the message RNA. It's the complementary bases. And if you can see here in this schematic from Wikipedia, you can see that the nucleobases follow exactly the same order as the non-coding DNA, and the key difference is thymine in the DNA will be replaced by uracil in the RNA. Both of them will be complementary to adenine, and they share the same piece of the molecules that bind to the adenine molecule. So basically for RNA, we have the four bases are cytosine, guanine, adenine, and uracil. When we are going to be reading a gene, the first thing that happens is RNA polymerase. It will actually open the DNA strand to start reading the coding strand. But how does the RNA know where to open it? Well, every gene always starts with the same 3 bases in order. TAC in the coding strand. It will be translated as AUG when the RNA polymerase starts putting the RNA in complementary messaging to the coding strand. And it always moves from the 5' end to the 3' end. It knows when to end because there's at least 3 different combinations in the coding strand that are translated as the stop codon. So when we have an RNA that's being synthesized, the RNA continues moving out from the AUG 5' start, continues adding the nucleobases towards the 3' until it finds the stop codon. So that's the way that the message RNA will be produced, the messenger RNA. Again, it will have the same sequence, or the sequence that reflects the non-coding sequence because it's complementary to the coding strand, which is encoding for every single specific gene. And what we get out is a messenger RNA that has the AUG at the beginning on the 5' end. It has one of the 3 combinations of the stop codon at the 3' end, and then it adds a poly A tail. So there's a bunch of adenines that are added at the 3' end. And this is very important for the cell, and it will be very important for establishing our tests for RNA, because it identifies what is the message RNA, which one is the RNA that contains the message. It's the one that has the poly A tail. When we have a patient and we do the nasal swab or the throat swab, we then use that cotton swab to put it into the media and we extract the RNA. So now we have the message RNA, we extract it, and oh gosh, what's happening? Oh, of course. I mean, we've got a ton of message RNA. That's what we have, when you sneeze or when you cough. So we're actually extracting a lot of RNA. That could be human, could be pollen, and of course, if the patient is infected, it will also have the RNA for the SARS CoV-2 proteins. It is included in all of the message RNA that we are getting from that nasal or throat swab. So it is there, but there's a lot of other message RNA in it. So this is the first step. We know it's there if the patient is infected. Now we need to detect it. The important thing for detection is that if we detect any of the non-specific, the patient could be having any other coronavirus. So the testing basically focuses on testing the spike protein RNA. So it's the RNA that encodes for the spike protein. So how do we do that? We're going to do a PCR and we'll do it together. Let me walk you through that. This is from Wikipedia, and what you see here is the poly A tail. So we have the message RNA that we obtained from the nasal swab, and remember, there's a lot of message RNA in there, okay? So the first thing we do is remember that the adenine in the RNA will combine with uracil, but now we're going to produce a complementary DNA of that RNA. That needs thymine. And because we have a poly A tail, we just add a lot of thymine, and that's the first step. We start producing a DNA strand that's going to be complementary to the message RNA. When we create this poly T tail, then we can add the other DNTPs with a reverse transcriptase. So normally, the message goes from DNA to RNA. But because we have an RNA virus and we need to create a complementary DNA, we use this enzyme that's called reverse transcriptase because it will transcribe the message from the RNA to the DNA. And that's why the poly-A tail is so useful, because by starting with thymine, with T, we know that we're producing DNA because we did not use uracil. We're producing DNA using thymine, and then we add all of the deoxynucleotide triphosphates in our mix, and they will automatically find their match, and they will start producing a strand of DNA that complements the message RNA that we got in the nasal swab. Again, a mix of a lot of message RNA. We're getting this in the test tube, and we're getting a lot of complementary DNA. So once we have this complementary DNA, we now finish the transcription, and what we get is a hybrid. It's a hybrid double-stranded. One strand is the message RNA, and the other strand is our complementary DNA. So this contains complementary DNA to all of the message RNA we got from the nasal swab. And I want to pay homage to Kary Mullis. You may remember him. He got the Nobel Prize in 1993 for the development of the technology for PCR. I guess that many people now take PCR for granted, but it really revolutionized the way that we can study the genes and that we can do, for instance, these testings, and everybody is acquainted with DNA testing or DNA amplification or PCR. So the polymerase chain reaction is an enzyme that can amplify the DNA using selected 3' and 5' primers. So what does it mean? As I mentioned to you, now you have in your test tube, in your mix, the complementary DNA to all of the message RNA. Now, because we know which one is the receptor binding site in the spike protein, we know the sequence of that particular piece that's very specific to SARS CoV-2. So because we know that sequence, we actually start with the primers on both ends. So we start the transcription using the specific 3' primer, 5' primer. We put the DNTPs in the mixture with the polymerase chain reaction enzymes, and the DNTPs will bind exactly, continuing the sequence from the 3' and the 5' end, will only amplify the SARS CoV-2 message. And that can be done again and again and again and again, because we have a lot of these primers, we have all the DNTPs in the mixture, and we are getting a lot of that DNA amplified. And because we use the specific primers for the SARS CoV-2, now the only DNA sequence that we can see amplified is the SARS CoV-2. And that's the way that we can get, just after 20 cycles, 1 million copies. So even though we began with a complementary DNA that had all of the DNA from the other strands, once we get the amplification with PCR, we are only amplifying the one that we need to see. If that piece of COVID-2 receptor binding site protein was not there, if the gene was not there, then it will not be amplified. So how do we know these primers? I'm just quoting the Nature Communication paper from 2020. This is the one that I found has actually sequenced and has provided, based on the differences between the proteins in the spike protein. It identified what is the forward primer, sequence on the 5' to 3' end, and it also published what is the reverse primer sequence, the one that goes from the 3' to the 5' end. So I don't know if all of the tests available are using the same primers, but these are definitely publications available so that people can use the adequate primers to amplify the sequence that will identify the COVID-2 genetic material. And it's, remember, a genomic RNA. We started from the message RNA in the nasal swab. We produced a complementary DNA because the amplification with PCR requires DNA. And so now we're identifying the complementary DNA that identifies the SARS-CoV-2 S protein for this virus. So what are the key points on testing? When we have nucleic acid amplification tests for COVID virus, there's many different ways to do it. This is just one example using the PCR. But what is key is that the routine confirmation of cases would be based using unique sequences of the virus RNA. So when we are able to use this kind of testing, this is the most appropriate. It's nucleic acid amplification with unique sequences for the SARS-CoV-2. If this amplification was done with sequences that are shared by other coronaviruses, then it's not a confirmatory test. You need to make sure the test is using the appropriate primers. And this is taken from the World Health Organization guidelines. But also another thing you need to understand is, again, for the RT-PCR from the Frontiers in Immunology, they published the importance of using specific primers. It is important to note, they conclude, that wrong use and interpretation could lead to disastrous public health consequences. So we need to understand what testing is being done, which primers are being used. Am I really going for the SARS-CoV-2 RNA? And that's what you need to understand. We amplify the DNA that is complementary to the SARS-CoV-2 RNA, and then you need to know which RNA of the SARS-CoV-2 is this actually amplifying. It should be the one in the S1 protein, or at least one that has been documented to be specific and unique for the SARS-CoV-2. Knowledge check. True or false? Let's take a look at the first 4 questions. Nasal swab test is based on amplifying the SARS-CoV-2 specific RNA. It's false because it's not the RNA what we amplify. We amplify a complementary DNA. Nasal swab test that includes detection of all 6 SARS-CoV-2 RNAs, a more specific diagnosis. Well, that's also not true. It's false because to be specific, we need to use the message RNA that's specific for the SARS-CoV-2, which is in the spike protein. The other 5 SARS-CoV-2 genes are common, and the sequences are shared and cross-react with other coronaviruses, so they would not be specific for the SARS-CoV-2. Reverse transcriptase from human infected tissue will produce the complementary DNA of a mix of messenger RNAs, including human and viral. That's true. It will include everything that we have in the nose and the throat. It could include microbiota and pollens and whatnot. So we produce complementary DNA of the mix of all the messenger RNA by using that poly-A tail and a bunch of thymines to start just producing complementary DNA to all the messenger RNA in the sample. Reverse transcriptase polymerase chain reaction from human infected tissue will amplify the complementary DNA of a mix of human SARS-CoV-2 RNAs. This is not true, it's false, because we amplify only the sequence that we're interested in by using the specific primers that correspond to a region on the receptor binding site that we know is unique. for SARS-CoV-2. If we move now to the antibody testing, I first want to draw your attention to timelines of what we know so far. So now that we got the message RNA and we went on to basically just produce the complementary RNA and amplify complementary RNA of the spike protein using our specific primers, when do we detect it? Basically, on the initial exposure, we start detecting it because remember, we amplify from minute amounts that are there. So as long as the gene is there, the genomic RNA is there, we were able to amplify it and detect it. And normally, by about 14 days, it is no longer detectable. What does this mean? When you do a nasal swab and it comes back positive with the viral load for the SARS-CoV-2, that individual is carrying the virus, and at least 14 days later, if not more, you need to double check on the testing to make sure that the individual now does not carry the virus anymore. This would be the normal cycle of life of the virus. It does infect the individual and usually it disappears by 14 days unless there is a serious infection and an individual could actually continue carrying the virus. That's why it is important to make sure that after you do the first RNA nasal swab test and it's positive, you wait for at least 14 days. You could even wait a month. Do the second one, and if it's negative, in some places, please check your local guidelines. And I'm taping this in July 2020, so I don't know when you're going to be listening to this, and better guidelines may have evolved a year from now. But if you get the second RNA test as negative, the recommendation is generally to repeat it, just to make sure that you're not missing anybody carrying the virus. So this is the time frame for having an RT-PCR nasal swab result that has been done specifically to amplify the complementary DNA that identifies the SARS-CoV-2 specific sequence in the spike protein receptor binding site. What happens to antibodies? Well, the curve of antibodies, and I'm quoting the Frontiers of Immunology published in April 2020 paper, is that we are going to have different types of antibodies. These are types or classes of antibodies. The IgM is usually the first one to appear in any kind of humoral or B-cell response. So when you have antibodies, generally you first are starting to produce what we know as the IgM antibodies, and they generally peak about 2 weeks after the infection. You can note that the first detection does not happen until the first week, whereas we can identify the genetic material right at the moment the virus gets to you. Your body has to generate antibodies, and that takes time. And for us to be able to detect it, it takes even more time. So IgM would be the first one to appear, followed up by IgA. The IgA type of antibody is an antibody very well described and characterized when we have pulmonary involvement. And we do know that this coronavirus has an avidity for the pulmonary tissue. That's one reason why IgA will start being produced and detected, but that takes a little longer than detecting the IgM. And finally, the IgG antibody, which is the last one to appear, and in this case, will show up about a month after. So all of the three mixes of the classes of antibody could be present in the same individual. They start being IgM, then IgM, then IgG. What the class tells us is basically a kind of a maturing of the immune response. Now, this is after about a month that the patient has been ill. We want to know eventually if these antibodies are going to stay with the patient. Are they staying a year later? What are the titers? Are they still there? And I just want you to know that in between this graph, between let's say the day 20 and the 1 year, you have dotted lines because that data is really not available. It's extrapolated. There's very few cases that we have who have had antibodies that are being studied and followed up for a year. This is July of 2020, and the disease is quite young. We still have not got to a year after the November 2019 first detection. So in summary, we really don't know. We don't know if a year from the infection, people who have developed antibodies will still have them. It is important to collect this data. It is important for epidemiological and scientific reasons to do the antibody testings, to produce more data so that scientists can understand how to interpret it. But it's not useful for diagnosis or for making any kind of decisions related to a patient's care. So for now, what we know about the antibodies is very limited. So let's take a look again at the Encyclopedia Britannica schematic of the virus. So we do know that antibodies to the specific receptor binding site in the spike protein are actually very specific for the virus because that's the region that contains differentiating features of the SARS-CoV-2. But also, this is the region, this is the protein that allows the virus to enter the host cell and infect. So antibodies to this piece are also what we call neutralizing, and they are protective. So having these antibodies helps a lot to the patient. Now, other areas, even within the receptor binding site or within the larger spike protein, can cross-react with other coronaviruses. So because it's cross-reactive, a lot of people could potentially have antibodies to other coronaviruses. We don't know if they may be protecting them against the SARS-CoV-2. We do know that the ones that bind the specific RBD sequences of SARS-CoV-2 are protected. But outside that region, we really don't know. We do know that those sequences cross-react. So the antibodies that bind outside could be binding also other coronaviruses. So patient could have those antibodies not because they had SARS-CoV-2, but because they had other coronaviruses. That explanation goes with the membrane protein much more clear. These antibodies would not be protective. A patient could develop antibodies to the membrane protein if they have SARS-CoV-2, but they're much less specific. So they could have these antibodies because of any other coronavirus. Same thing happens with the envelope protein. These are not very specific, although they could be generated by patients. For the nucleoprotein, a similar situation than other regions of the spike protein that I found could potentially be cross-reacting, but also they could neutralize because they are going to bind the nucleoprotein that normally would help the virus when it's actually amplifying itself in a host. And by not being able to introduce a nucleoprotein, they may be protective, but they are also cross-reactive with other coronaviruses. Take a look at the spike protein because that's the one that basically is more specific and can differentiate this SARS-CoV-2. This a paper on viruses published in 2020 looked at the structure, and I just want to let you know that we are looking here at the SARS-CoV-2 protein. So this is the coronavirus for SARS that we know. okay? And what they did was to overlay the SARS-CoV-2. When they looked at the SARS-CoV-2 epitopes, what I want you to see is that they have identified in blue sequences within the receptor binding site of the S protein. You can see all the blue pieces in there are epitopes or pieces of protein that are non-identical in the SARS-CoV-2. This means they are specific for SARS-CoV-2. This is a very important concept because if we have antibodies to the blue epitopes, that is undoubtedly driven by SARS-CoV-2, not by the other coronaviruses. If we have antibodies to other pieces of the spike protein, they could have been from a SARS-CoV-2, but they could have been also from other coronaviruses. So these are the more specific pieces. Let me walk you through the assay. Individual will donate blood, and this blood is collected, and we get the blood that we're going to test on what we call a platform or an assay. So we need to test for antibodies to coronavirus. So we use the coronavirus on the assay, or we can use the whole spike protein, not all of the coronavirus proteins. We can actually just use the receptor binding site, or we can go for those blue epitopes or blue pieces in the receptor binding site that are specific for the SARS-CoV-2. Different companies will develop different assays, and these assays can be done using different technologies, but each one of them needs to provide the specifics of what exactly are you testing. So let me walk you through these 4 different options. There may be more, but these 4 examples in the case that a patient was exposed to the SARS-CoV-2 and now they have antibodies. So in those droplets of blood, there are antibodies. And when we incubate these antibodies on the platform that we're using, if they have the antibodies to the COVID-2, they bind to these particular proteins on the assay. Then these assays have a detection method that varies according to the different assays. And this detection method will actually identify if the antibodies are bound to the protein that's on the plate. If the person did not have antibodies, these detection methods will not bind to the plate. They bind to the antibodies that are bound to the protein that's on the plate. So how do we interpret true positive results? And I want to direct you to my other video that's probably easy to find. It's called the Devil Interpreting Test Results, and I did it in June 2020. So let's look at the first assay. We use the whole virus, and a patient may actually have antibodies to the whole virus. So in this case, it does cross-react with other coronaviruses, and it may actually cross-react with other viruses, because we don't really know which piece of the virus or which protein of the virus this particular antibody is binding. So honestly, if a patient has SARS-CoV-2, this test will pick it up. But if they don't have SARS-CoV-2, they have another virus, it could also be considered positive. So in this case, this particular assay is going to be very sensitive because it is sensitive to picking up any kind of antibodies that you have there, but it's not specific. If a patient had other kind of coronavirus infection and mounted antibodies to them, then this is not specific for SARS-CoV-2. When we look at the next example, now we use the whole spike protein. Now, using a spike protein discards immediately a lot of the other coronaviruses, but it really does not differentiate between the pathogenic coronaviruses that we know have caused disease in humans. It is also cross-reactive, although not cross-reactive to everything. So it is a bit less sensitive because it will not pick up every coronavirus infection. That's why I used a font that's slightly smaller for sensitive, and it is slightly larger for specific than the previous example because it has less sensitivity than the previous example, but it's a bit more specific, but not quite as specific as we would want it because it would pick up, again, antibodies from every coronavirus that shares whatever the spike protein piece we used in the assay. The third example uses only the receptor binding site of the spike protein. The specificity is going up because the receptor binding site is a highly conserved area for the pathogenic coronaviruses, but it is less sensitive because it will pick up less of the other coronavirus antibodies. When we look even further to the assay that now just used the blue regions, the ones that are unique to the SARS-CoV-2, now we get the most specific assay. But it doesn't have much sensitivity. And why do we care about this? Well, the antibodies to the virus are idiosyncratic. Each individual will produce the antibodies according to their own, not only the genetic background that they have, the environment, what they eat. If you eat more protein, or if you eat more vegetables, or if you have a kind of diet that's different, or if you were recently exposed to another virus, your immune system responds differently. And your immune system could be identifying really any piece of the virus when you get infected. So if your body is actually infected with SARS-CoV-2, but happens to actually bind a protein that's not in the spike protein, that doesn't mean you were not exposed to the virus. So not being very sensitive will produce some false negative results if what you want to know is that the patient had the virus. For now, in July 2020, if you want to know if the patient is infected, you need to do the nasal swab or the throat. You need to use a nucleotide amplifying test. You need to use a PCR testing. You need to look at the genetic material of the virus, if it's present or not. Antibodies are currently not useful for diagnosis because we don't know exactly which is the best way to identify them, when to identify them. This is work in progress. On the FDA website, you can find the FAQs, the facts and questions, on testing for SARS-CoV-2. And this is the piece that specifically refers to the antibody tests. I refer you to the most current version that you have at the time you are watching and listening to this video. But what they basically are saying is that the test for antibodies cannot be used for the diagnosis of infection. They also make sure to say it could aid identifying individuals that have an immune response, which means they had either recent or prior infection. We do not know how long these antibodies are going to remain in the blood. So we do not know for how long these individuals will have the antibodies. So for now, it's a tool for understanding the epidemiology and the science of the human response to the virus. But it's important to know that if you have a negative antibody test, it does not mean that you do not have active SARS-CoV-2 infection. You need the RNA test for that. On the Centers for Disease Control and Prevention, they have a updated guidelines, and I'm using the ones that I have more recently updated. They are from May 28, and this is referring to the antibody tests. So the message is clear. They are not going to tell us if a patient is currently infected. A positive antibody test does not tell us that the patient has the virus. More importantly, there's not enough information in 2020 to understand exactly if this immune response is going to be protective. We don't know that. All we can say is that the patient has an immune response to the virus. So, bottom line, at present, we need to use caution when interpreting test results. This is for antibodies. Okay, let's go over the last 4 questions of the knowledge test. The reverse transcriptase polymerase chain reaction test identifies SARS-CoV-2 RNA up to 14 days after infection in human tissue. This is true, it's what has been described so far. Of course, in individual cases, it could be 14 days plus minus, but in general, 2 weeks. And that's the reason why a repeat test is at least 14 days, if not more, after the first test. Let's talk about antibody testing. Anti-SARS-CoV-2 for diagnosis must detect the receptor binding site and neutralize the virus to be confirmed positive. I hope that you now realize this is false. The anti-SARS-CoV-2 should not be used for diagnosis, but if we want to follow up a diagnosis, they could have had different antibodies to the spike protein or even to other proteins like the nucleoprotein. So for a follow-up on this diagnosis, they do not need to be directed to the receptor binding site and they do not need to be neutralizing. This particular investigation of which antibodies from which individuals have neutralizing capacity are being used today for asking these individuals to donate blood, and it's one first line of potential treatment that will be more of an emergency treatment using antibodies from people who are convalescing and whose antibodies neutralize the virus. This is being used nowadays. It is a limited therapy because it's what we call passive immunity. It does help the patient tremendously, but it does not protect the patient long term because these antibodies were not produced by the patient. They are from another person. They are helping the patient beat the virus because it neutralizes the virus, but it does not convey protection. Has its uses, and if you are discussing treatments, you need to understand the pros and cons. Anti-SARS-CoV-2 test must not cross-react with other SARS-CoV-2 or MERS to identify specific COVID-19 antibodies. This is true. In research, it is basically used if we want to identify specific COVID-19 antibodies, again, for research purposes, then the test should not use a platform that uses proteins that can cross-react with other coronaviruses. So this is important for understanding what is the kind of test that you're getting. If an individual is getting a result of an antibody, understand what was tested and what exactly is this telling you. After the reverse transcriptase polymerase chain reaction confirmatory test, anti-SARS-CoV-2 antibody testing used for follow-up identifies immunological memory and protection for at least 1 year. Well, this is false because it does identify immunological memory, but it does depend on the antibody testing that was done. The anti-SARS-CoV-2 antibody testing tested something that is actually not a neutralizing area, then it will not provide protection. And also we do not know that even if an individual has neutralizing antibodies, we do not know for how long they stay in the body and how long this is going to last. All of this information is extremely useful because this is going to create the basis for understanding once we have a vaccine or several vaccines available, we will know which kind of antibodies we are mounting after being vaccinated, and we will know how often we would need to get a booster vaccination. We all know that for the influenza, we have to get it every year because these antibodies will not provide protection after a certain amount of time. So for the COVID-2 testing, we need to understand what is the dynamic of the antibodies. Are they still neutralizing after a year? These are all questions that we don't know. So for now, we don't know that they provide protection. To create this video, I actually used several different references. I encourage you to read more, make sure that you are up to date. This is a very vast area of growth in terms of scientific knowledge. And I just hope that you enjoyed my video. If you got to the end, I hope that you like it. And if you want, check other videos on my channel. Thank you very much.